Hi, Bern. How you doing?
Does it? Oh, yeah. Oh. I was going to make a joke about the first who come up can sit and the other must stand, but now it's we are all set for our discussion. Let me introduce to you the people who will be participating in this roundtable. This roundtable will be on the question of what do results from clinical trial mean. And we are going to have arguments which we can take from what we have heard now and also what we have heard previously in the difficulties of some studies, but we are not going to discuss details on the particular studies, rather to take the argument to have more a general discussion in which we like you to please participate. So I like to introduce a question in which direction should I go? Ricardo Dolmetsch is here, so he's here. No, he's not. Oh, he's coming. Great. Oh, this is the one who didn't have a chair. Well, now I understand. Ricardo, you have heard him speaking, is from Unicure. We have Irina Antonijevic from Triplet Therapeutics. And we have Peter, whom I don't need to introduce again, from Roche. And then we have Henrik Zetterberg, who is a group leader of the UK Dementia Research Institute on fluid biomarkers for neurodegenerative diseases. Then we have Jessica Koehli, who is working with us in Bern at Siloam, who is both from a family with Huntington's disease and also working as a study coordinator. Then we have a lot of chairs or past chairs and so on from EHDN, whom I will not introduce, but with so many chairs, co-chairs, and past chairs, I hope you will behave very correctly. So let's start the discussion, and we have prepared together some questions or some ideas, but I would like to have this discussion be very vivid, and I don't think we need so much light here, but it would be good to have lights also from the floor, so we can see if people would like to discuss questions. So one lesson which I think we should discuss, or one point which we might want to discuss, is a lesson from the Roche trial. Let's start with this one, where we have the situation during a study with a planned examination of the data. The planned consequence to it is to stop providing the drug. So let's maybe discuss already the matter of what is the situation here. Would you like to comment on this first part here? And where is the coach?
Sorry, I didn't fully understand the question.
The question is the discussion about the planned, what to be done when this happens, that the data safety committee would see that difference.
I mean, I think there's no overstating how devastating it was for the community when the news was released. I think what's a real testament to everyone who was on the study is that people still stayed on the study even after that news. And I mean, that's why we were able to share all the data that we were able to share this evening, because of that dedication, we now know what happens in the post-treatment period in terms of the clinical and the ventricular volume. And if it hadn't been for the patient's dedication and commitment, even after that devastating news, then none of that would have been possible. So I think that can't be overstated.
Bernard, you would like to comment on this question too?
Sure. I think it is critical for each clinical trial that there is such a thing like the Data Safety Monitoring Committee that would alert, because they have access to the unblinded data, of problems. In this case, as we all recall, Can you still hear? Yeah. And there was a situation where one dose arm clearly was inferior to placebo, and one dose arm that was equal to placebo. Of course, disappointing in that no improvement was seen. I think this is an example where one could argue what decision the DSMC could have taken. They could have, for instance, recommended to discontinue one dose or exposure arm and recommended to continue with the one that disappointingly showed no difference in favor of the active treatment, but equally did not result in an unequivocal safety trial. These are discretions and various options. We know what the decision was at the time. I can understand the reasons for this. At the same time, it has implications because we don't know whether a continued treatment would have yielded a different impression at the time. So it illustrates how important these sports are. It also illustrates that these decisions are difficult for the participants, for the companies, and it raises the question with hindsight, and there's always 2020 vision with hindsight, whether there would have been other options.
At the same time, it's also giving a sense of safety, security, that we know that these mechanisms are being put into place. So we as investigator can have that trust, and also, of course, the participant. Which leads to a further question, but before I do so, I would like to ask you if you have remarks or questions, or who would like to participate in the discussion of the different topics as we go along, to please raise your hand and you get a mic, and we can introduce you also to the discussion. Now, the next point is another one, is considering now what happened in another situation, which was the study by Novartis. So they needed to stop also, or to pause the continuation of the study, because what happened there, they had looked at very intense signals in terms of safety along the line of the study, including possible side effects related to peripheral neuropathy, including also precise measurement of that, like of a kind of a biomarker. They also included NFL, etc. So here we have another situation which I would like also you to please comment on something which had been embedded in the protocol and actually then occurred. Maybe Henrik, you would like to comment on that in terms of
yeah, I could just mention that when listening to the presentation now also, and what happened with the Novartis drug, it's so important to have several biomarkers, both imaging and fluid-based biomarkers, because then if one looks at these data and several of these markers change in a manner that does look a little bit unfavorable or scary, then you're so much more certain that something is going on which might not be good. So I would really warn against taking decisions against individual results, but look for patterns. That is my comment on this.
Again, here the previous data told them that they should be very careful in that precise element. I saw a question over here, and you please. Can you check the mic with her, please?
Hello. Okay. Yeah. Sorry, we just wasn't on yet. So going back to the point I raised earlier, is there anything we can infer about, so let's take the hypothesis that was presented by Peter, that there's a loss of turnover of CSF or a decrease in turnover of CSF, and is there a Huntington function that we can pin that type of mechanism on that could then be considered in future trials?
This is a good question in terms of using the data to then inform future trial. Would you like to comment, Peter?
I think that in terms of understanding the mechanism, I guess we can only go so far in terms of looking at relationships with fluid biomarkers and ventricular volume, as we've done. There are other approaches in terms of looking, using MRI approaches to look at CSF flow and stuff like that. I think the difficulty is that those techniques may not be in a place where they're ready for clinical trials, and trying to do that at a large trial across multiple sites is very, very challenging. And so we're a little bit limited from a trial perspective in terms of what we can learn.
So I don't know if Fred and Sondre
maybe
are still
again,
here.
I would like to stress the idea not to go into particulars of the studies, but
No, but
in
I'm
terms
talking about the
generalities.
pattern that might arise again, that the decrease in turnover in CSF is related to the loss of wild-type Huntington function. For example, what about ciliary function? Wouldn't that potentially have an effect on CSF flow and turnover?
Yeah, so there is a relationship with cilia and wild type, but I think there are no biomarkers that I'm aware of that we could use to inform those specific questions. I think it's challenging to do that sort of outside of preclinical or animal studies. I wish we had biomarkers that could better understand it, but I'm not sure we do at the minute.
I guess one thing I might just add to that is that so far the only systems in which you can look at it, we really haven't seen big effects of knocking down wild-type Huntington on CSF dynamics. So while it's possible, it's more likely actually that it has something to do with off-target toxicity, I would say. And for what it's worth, other ASOs are known to have problems associated with CSF turnover. It seems as if that's a more plausible explanation. Obviously, it has to be investigated further, but it's hard to investigate in humans. I think this is the sort of thing we're going to have to do preclinically.
Christina?
Thank you. I have more a comment than a question, but is related with the previous discussion of the DSMBs. I wanted to make the following comment. So first of all, like Bernard said, the DSMBs are a critical piece of any clinical trial, and many, many times, not always, but many, many times, they are a regulatory requirement. So the trial cannot be authorized if does not have a proper DSMB in place, and the composition of the DSMBs and the structure, the charter of the DSMB. So there is a number of regulations that inform the composition, the role, how many times they have access to the data, which type of data they have access. And I must say, unblinded data is not always the rule. So, it's something that sometimes is pre-specified in the charter, is something that sometimes happens because of the need. So that's one thing that is very important that everybody has in mind. The other issue that are the concrete examples that we just discussed, they are in a way fortunate as a, let's say, learning process, because they typify two things that happen in clinical trials. In the Roche trial, there is an event that was unexpected, completely unexpected, that would be a higher, let's say, higher rate of deterioration, at least in one arm. And I must say, that's not usual. Even today, I have been discussing the difficulties of neurodegenerative, of drug development in neurodegenerative diseases. And as all of you know, the majority of the difficulty is the proof of efficacy. Alzheimer's trials fail because of efficacy. Parkinson trials fail because of efficacy. It's not so common that they fail because of safety. There are other diseases where they fail about safety, mostly in cancer. But efficacy is the most common reason. So this situation that happened in the Roche trial was unexpected, and it's one of the reasons why DSMBs exists. I must say we could discuss at length, but I agree completely with the decision of stopping both arms, given that was a safety issue, and Bernard argues we don't know what would happen with the other arm. Yes, we don't know what would happen either for the good or for the bad. And we always work on the side of caution. The Novartis example is also a very good example because it is an expected side effect. The preclinical data and the pharmacology of the drug predicted that this side effect could happen. What was unclear was how frequent it will be and how severe. But the effect itself was already known. And again, the decision to be made was, did it cross a threshold that is not anymore tolerable? And I have not seen much data, but again, I think the DSMB made a proper decision. So I think the more we see about these type of discussions, the better we know how these different, let's say, scenarios pan out. My final comment is resonating a bit with the concern that Bernard also has. There is a huge literature about ending trials because of futility. And this is a completely different issue that is unrelated with DSMB. It's a strict company decision. Typically, companies plan interim analysis to close trials that don't have a likelihood of meet the endpoint in the long run. And we can sympathize with the idea because, first of all, patients are not exposed for so long in a clinical trial that will never get to the result. Companies will not spend all that money. So there are good arguments. But there is also an extensive literature that says that sometimes there are missed opportunities, and if those trials have gone to the end, there would be data for the community to build a better research in the future. So there is a lot of controversy about ending trials for futility, but it's a different thing from DSMB decisions. So that's what
Thank you. These are also based on data analysis, but Ralph, a short comment on the same topic?
Well, one short comment. I just think in the Novartis trial, one of the interesting things was that we had a biomarker, if you wish, a clinical measure and a biomarker change. So we had the nerve conduction velocity changes, and actually we had clinical changes as well, which I think pointed to having a look at this a bit further. But unrelated to that, I would like to ask you all, discussing like the multiple clinical trial approaches that we currently luckily still have in Huntington's disease. And one take-home message from the Roche trial may also be that actually I think we moved very quick from phase 1 to phase 3. Is there a concern in the community? I do have it, obviously. Is there anything we can do to, as a community, as the EHDN, as the investigators, guide pharmaceutical companies, guide the field in terms of what trials do we actually endorse at what point? Because we might have limited resources in the end of the day of early stage patients. I think it's important for us to try to digest what guidelines, what critical decision points can we use in the future as EHDN, as a community, to say, okay, we endorse this trial in this size now, or maybe provide more data, do a phase 2. Where are we there? Are you also, every one of you, of the clinicians, maybe also the sponsors, concerned that we might end up in a point where we might not have sufficient early stage patients, because that's where the whole field moves at this point, to do meaningful trials? Because I think it was different 5 years ago when we did small molecules and people could actually be kind of recycled to the next trial, Whereas now, when we do Unicure trial, for instance, obviously people receiving gene therapy are no more able to join other trials, and the same is true with ASOs as we learn it right now. What do we need to be still in the position as a field to conduct phase 3 clinical trials?
Thank you, Ralph. The point is very important, and EHDN does endorse clinical trials. And maybe Anne, you would like to comment on that very important question?
Yes, thanks, Ralph. I think there's several questions in there actually. But so the issue of trial endorsement is really important. I mean, I don't think EHDN should be a gatekeeper for trials. And what we should be doing is really promoting discussion based on these sorts of issues. So these sorts of discussions amongst ourselves with the with industry, you know, I think what we could say is finding more platforms to have these sorts of discussions is really important. And, you know, clearly over the last few years, our understanding of clinical trials and what's achievable and what we need to be thinking about has evolved massively. So I think this is a collective effort, and I think, you know, I think it's an important point you raised, but I think it needs, you know, more intense dialogue to move forward.
Thanks. Michael, yes, on the same topic, another comment?
Well, it's sort of on the same topic. I'm just curious to hear the panel's view. You know, when we think about treatment of Huntington's, but also other neurodegenerative disease, this is going to be long-term therapy, maybe 20 years, hopefully 30 years. And I'm just wondering about the thinking about the thresholds for safety as we think about the implementation of clinical trials, about how that alters our thinking. Very different to cancer, where you're going into treating short-term. Here we're treating long-term for decades. And related to that, I just wonder from Peter, even though that the safety board didn't require you to follow long-term these patients, I wonder if that's still something, because I think that would also be helpful for the community to have ongoing follow-up on those patients.
So definitely this is something that we're looking into at the minute. I think further follow-up through some pathway would be very useful to understand the longer term effects. So it's something that we're actively discussing at the minute.
In terms of long-term treatment, the approach by Unicure to do a one-shot treatment for a long time is quite a good example on that. Would you like to comment or discuss a little bit about another issue, another situation here, which was a severe side effect which occurred, which would not actually directly planned in this way, like the neuropathy? Would anybody like to comment? Maybe Ricardo yourself?
Absolutely. So, I mean, let me just think about this. So I think, you know, what we observed, or at least what we feel we've observed at the moment, is sort of the equivalent of an infusion reaction. You know, out of 14 patients, 3 of them had this symptomatic edema in the area that we infused, and it seems to be related to the concentration of the virus. It seems to be self-limiting in at least 2 of the 3 patients, and in the other one it seemed to be treatable. And we feel that there is probably a way of getting past this at the high dose. Certainly the low dose was very well tolerated, and the low dose seems to be effective in reducing Huntington. So moving forward with that would be viable. But I think it might also be possible to move forward with the high dose if we monitor people really carefully, and if we think about treating with steroids when necessary. But this is something that the DSMB will have to decide. And we're in the process of collecting all the data to make completely sure that that's the right thing to do. Just more generally, when it comes to long-term monitoring of patients, of course, in gene therapy, you're treated forever, which is both an advantage and a disadvantage. The main advantage is that, well, we will continue following these patients for at least 5 years and possibly longer. At some point, even if we don't treat that many patients, we ought to know whether there is an effect, because you would expect them to progress. So in a sense, you can get a lot of data if you're prepared to wait with the gene therapy. On the minus side, of course, you know, you're treated forever, and so that does preclude you from getting other therapies, which is a potential issue. So, I mean, I guess the position of our company is that one-time treatment has the potential to be transformative in medicine in general and perhaps in neurology, because it does get past the compliance issue and so on. But we'll just have to see. I mean, in the end, I think the issue for all of us is can we get something that works? And then the other part we can kind of manage.
Thanks. Bernard, a general comment?
Yeah, I would like to address the question that Michael raised, long-term toxicity. I think the way various DSMCs and companies reacted to concerning signals was a very judicious one. Pausing application, observing what is happening. If you don't know what is happening, how to interpret a signal, I think that's judicious. And it's a reality. You alluded to it, that we may rather rely on patterns than on single values to come to a sound judgment as to whether a signal that concerns us is of real implication or not. I think it's judicious to consider pausing. It's not the nicest thing for the development of a compound, but I think to me, it makes sense. Now, to the core of the question, I guess, there's always a question of risk and balance. HD is not a benign disease. There's no question about this. It's what trials, to my mind, can only do is provide signals as to the risks, and then it's up to physicians and patients jointly to decide what is an appropriate risk-benefit option. But it's unlikely that this is an answer that can be conclusively addressed in a clinical randomized controlled trial, but there are other mechanisms and follow-up to address this.
Henrik, would you like to add something about the biomarkers in that question here?
Well, I wanted to quickly say something which is like actually outside of clinical trials, but I think electronic health records, like, I mean, it's not only about, you know, a sponsor will not follow a patient for 50 years, or maybe not, most won't. But I think as a society, I think it is really important to have a tool to do data mining, even when the drugs are approved, to try to identify signals, because only when more patients are taking it, we might pick up on something. And so I think it's, and I'm saying this because obviously I now live in the US, and I do think that it is sometimes quite astonishing how health records don't talk to each other, as, you know, change the insurers or so. So it's sort of something that we cannot solve as an HD community, but I think we can at least sort of advocate for that. In terms of biomarkers, so I was thinking, as we were talking about NFL and when Henrik mentioned that, I think it was also a Roche presentation where there were some issues with the assay. And, you know, this was presented at the CPAS consortium, so I think we cannot sort of share all the details, but I think having confidence in the biomarker assays, and we have actually experienced this ourselves, that we couldn't just generate data and then add on data, but we actually had to basically create a new baseline. So I think as we select biomarkers that we use for signal detection, I think we have to be absolutely sure that we are really confident that they sort of measure this reliably and that we know what we base our decisions on. Because I think if this is not done, I mean, you can't think you have a signal and you actually don't.
So, Henrik, the good biomarkers.
My comment on this is that one, of course, really needs to be careful with saving samples throughout the study so that samples can be rerun on the same plates, because also with very well-validated assays, there can be slight drifts that can be misinterpreted as some type of treatment defect. So that is really important to keep the aliquots in a biobank so that you can, in the end of the study, do everything side by side so that you never will have to consider this aspect. And then from the Alzheimer field where I've been a lot, there have been studies that closed because of safety issues. The AN2092 trial, the BACE inhibitor trials with cognitive worsening, and then also a couple of other trials that really also had signals that were not good. And then it became super important to do this type of long-term follow-up post-drug exposure. And AN2092 also had one arm when actually some patients came to autopsy, and the A-beta removal by the active immunization could be verified, which was super important. So my next suggestion to all the companies is that when a study is cancelled, make all samples available, if it's okay for the patients and the study participants, of course, and make the community examine them. For the BACE inhibitor trials, Alzheimer's Association formed a little bit of a crisis group involving a committee of researchers not involved in the studies to try to troubleshoot why BACE1 inhibition gave cognitive dysfunction at a higher rate than expected. And now the consensus of that group, it's not a consensus yet, but the data indicate that BACE1 inhibition led to APP accumulation in the pre-synapse, giving you a reversible synaptic dysfunction, which is so important to know. So that's what I would like to say. That was thanks to some companies sharing samples, sharing a lot of data. So if a company stops a drug, I think it should be mandatory to make everything available. If it goes well, of course one has to consider what the study participants agreed upon. But that could also be made clear from the start. And I think most study participants would love such a part of the informed consent document.
Sharing, this is a very important word. I'd like to continue on the discussion, sorry. Sharing is something very important, and sharing, there is a new aspect which actually didn't realize, which we have heard about the markets. So there is a market, and there is money, and this is being shared. And Irina, you told us about difficulties that your company has been facing. You mentioned that before in your previous talk. So would you like to comment more on that? And someone else would like to comment on that too?
So specifically about sharing samples?
No, sharing the market which
okay.
Companies need to share, and if
sorry.
the results of some trials are not so good, and if it occurs at several occasions, that the market is less interested in fostering HD research.
Yeah, so, I mean, this is, I don't know how to address this, but I think it is so impossible to conclude from one study what could happen in another study. I mean, even if it would be the same, let's say, even if we would have had the same approach of HTT lowering, these are different molecules. So I obviously cannot educate the analysts or the investors, but I think what we see, I mean, we see it in both directions. I think we have also seen, I'm going here out of a limb, like in the Alzheimer field, like how everybody does the same, and then one trial after the other doesn't yield the result, and how there doesn't seem to be a solid rethinking. On the same side here, it was maybe a situation where there was too quick a concern about this general field. So I think it is something that we maybe as scientists or as a community have to continue to explain to people who are less into the weeds of what we are doing, that these are different mechanisms, that these are different molecules, different routes of administrations matter, and that we should not consider a field of not worthwhile or too risky just because we have something that didn't go according to expectations. I mean, Christina said, I mean, there are so many trials that don't go according to expectations. So we cannot assume that in a field that has now seen this really plethora of trials, which is wonderful. It's wonderful to have the opportunity. So we have to be patient and give ourselves time to understand what is happening, understanding the complexity of the disease. But I also do want to say something about sample sharing, because this is really on my mind a lot, and because we have just gone through Shield HD, where we changed the informed consent. We wanted to make sure that there is a sort of broad access to the data, but this is something that is also not always easily accepted by all IRBs or ethical committees. So, like if you have a very, very broad research scope that we would need in order to be able to use the samples for some future research. So I think, again, there needs to be a communication at the institutional level of those ethical boards and IRBs, that they would accept this kind of language, and also explain then to the patients and the participants, again, that this will not be used easily, but that there is a clear rationale for using it. So I think making the HD community really part of understanding of what could potentially, how these samples could be used in the future.
Thank you. So, Jessica, just a second. I was told that there are many, a huge number of questions, but they must be very, very short because the time is running, and my Swiss clock tells me another 10 minutes, but then it will be stopping. But it's very interesting, so please.
I have a more general question on how to manage participants and patients once a trial fails. Because I feel like we as researchers, we do know a lot of our experiments don't work out, and we have a much good understanding of that. But the general public doesn't know that, and I think especially for the past few trials, expectations were incredibly high within the patient community. And then it didn't work out, and it had a massive emotional fallout for those patients. And my question is more, will there be a change in communication with our patients and our participants to manage hopes and potentially the emotional fallouts of what happens when it doesn't go to plan, basically?
Yeah, maybe Jessica, your comment would be on that direction also. As a coordinator, we needed to get in touch with our people where you said we stop the providing of the drug. So, Jessica.
Yeah, well, it was difficult. We were the bad guys breaking the news to the family, but going back to what she was saying, as a family member, when I told my family that the study got canceled, one of the comments was, if Roche said it's not going to work, then nobody is going to do anything else. So that was like a shock. And I'm coming from a family that is not very open to talk about this disease, the less the better. But I do think that we cannot make a conclusion for just one study. We need to keep on going. And talking about a study coordinator, it was a lot of work. Again, we were the bad guys, not only for the family members with the patients, but also with a lot of the staff members. We had to disorganize everything, to again organize everything, reschedule appointments. And most of them have their own requests. Oh, I want to have that tomorrow, at this time, or today. And at the same time, we got the monitors here saying it has to be now. So it was very difficult. And I think it was a challenge, but here we are.
So another question from the East.
Hi, Lauren Byrne, UCL. I kind of feel a bit conscious that I'm holding up us from drinks, but I thought a lot about all we are learning from these trials, and I think we really need to commend, I think, Roche and Triplet and everyone, Unicure, for their commitment to really understanding why these went wrong. It seems like we are understanding there are a panel of biomarkers that might be useful for making decision-making earlier in clinical development, particularly in that phase 2 dose-finding phase, which, as we know from hindsight, we rushed. I think rushing from phase 1 to phase 3 with Roche was probably not the best idea, but everybody wanted that, at the time. So I think anybody that's can say now that that was a stupid idea. We were all supporting that, I think, to the most part. Could we start thinking about designing trials that are a bit more creative and adaptive to include cutoffs of these panels of biomarkers that could then, instead of stopping the trial or stopping the arm, that then signals a change in dose to a new arm, similar to like multi-ascending dose or adaptive trials, but at a more phase 2 level?
Well, I mean, the idea of, running adaptive trials, I think, is something that many companies are considering. I think the issue that we have with biomarkers is that we don't have good enough validated assays. So they're not really ready for clinical trials. So there are a whole bunch of things that seem plausible, but basically everybody uses NFL and mutant Huntington. So the only two assays that are really genuinely, that are good enough to be able to make any decisions. And so that's very limiting. I mean, I think once, for example, if we had a PET ligand for Huntington aggregation, if we had a way of looking at actual loss of medium spiny neurons, then I think we might be able to make better dose decisions. The game, it's not a game, but the strategy in clinical development is being able to make decisions as rapidly as possible with as few people as possible so that you can set yourself up for success. And in neurology in general, we just don't have the tools. So we just need better markers to do that. I mean, I completely agree. Adaptive is the way to go, but the practicality of it is that you can't make decisions because you don't have the decision-making tools.
Thank you very much. The co-chair didn't have a chair, so he couldn't chair. This is really bad, and I had felt bad the whole time. But we are going to conclude, and you will give some remarks, of course.
Yes, so I can. So thank you. That was great to watch from the audience. Probably just as exciting as watching from up here. And my new calling is as a chair of housekeeping. So I have a few housekeeping announcements to make. There is the social event is going to happen now. So the people of you, or the members who have this red dot on your name badge, are cordially invited to go to the past the registration desk to the social evening with drinks, finger food, and a DJ, I hear, and have a look at the photo exhibition that we have. Thank you very much.
And also, I would like to thank you and thank the intervenants for the discussion, the preparation of that discussion. And it's a challenge, but I think also the participation of the public. So have a nice evening.
And, those people should get there. For everybody attending the networking event, please get over there as quickly as you can so we can start the activities. We'll be celebrating the 10 years of Enroll HD, and we'd like you to be present for the presentation and enjoy the treats that we have planned. Thank you.